let's talk about typical DNA gels, how to present typical DNA gels that you're going to see in publications in figures. So this is an agarose gel. This is the color original picture of it, and where you're seeing these bright pink lines is a representation of a plasmid DNA. So the first thing that I'm going to do is I don't want it to be crooked, okay? So the first thing I'm going to do is I'm going to straighten it out. And to do this, you'll notice that when I imported this picture, it comes into one layer and it's locked. So I'm assuming I have to unlock it. As soon as I unlock the layer, now the transform controls show up as long as you have this box clicked. Show transform controls. Okay, so the first thing I'm going to do is I just want these lanes to be horizontally straight. Okay, and I can test this by taking the box tool and just kind of making a line and seeing if it looks straight. It looks straight to me. That looks good. Next thing I'm going to do is usually agarose gels are presented as black and white data. That's pretty common. It's also in general when you're working on scientific data, and if you can present it in black and white, it's usually a good idea to opt for the black and white because most people are not printing in color. So when they print off these manuscripts, it's really good idea that you sort of just inherently program your data to look good as black and white. So I will always make my agarose gels, my DNA gels, black and white. So we're going to go to image, mode, grayscale. There we go. Now it's black and white. Now the next thing is once I make this black and white, you can tell that this would be hard on a printer. Again, thinking about making things easy for the person who has to print this off and making it easy on the eyes. One of the things that you'll notice is most of this image is either gray or black. It's only the data is very small and the data is the white part. So we can actually do a transformation here, which is called invert, which is not changing any of the data. It's just literally taking the reciprocal image in terms of the black and white. So I'm going to do image, adjustments, invert. And now this is most commonly how agarose gel data, DNA gels, are presented, as the bands as black and the gel as white, such that it's actually easier for you to see the data. It's much easier on the eyes, easier for you to see what you need to be looking at because it's dark black, whereas the background now kind of fades actually back into the background. So let me just show you before, after. This is most commonly what's done. Now, keep in mind how you present your figure is going to be entirely dependent upon your data and what you're essentially what is pertinent. Okay, so within this image, obviously, I think we would all agree that there's nothing that's pertinent outside of, say, like this region. I mean, you might want to include a full ladder. If you don't know about DNA, these bands are marker sizes, so I know how large these bands are in terms of size of the DNA string by comparing them to these markers. So certainly we would all agree that I could cut out everything outside of this box and not lose any information. Okay, so I'm going to do that. So once I have that box selected, I'm going to do select inverse, and I'm literally just going to erase the stuff outside of the pertinent information. Now what you'll notice is now I have erased and there's nothing underneath. There's no layer underneath. So to make that white again, I'm just going to do layer, new, background from layer. Oh, I didn't want to do that. That just essentially converted everything on the outside to white. I don't want to do that. Let me do layer, new, layer. Sometimes I'll bring it underneath. And now I can paint a white layer underneath. I'm just sort of walking you through the decisions that I do when I'm, oh, shoot, the opacity is low. I can finally get it to work. Okay. So now what we have is a layer with our gel and a layer with a white page underneath. Now, most commonly what people will do is, what's very, very common is to construct a little black box surrounding the data. So let me show you how you would do this. You would make a box. Once you have the box which borders the data, you're going to say edit, stroke, color, black. And I'm guessing here, I'm not sure exactly how many pixels, what this is going to look like, but 5 pixels. Let's see what it looks like with 5 pixels. So you can see at 5 pixels what it did is it made a little tiny black box around the data. I think that's actually too thin. So I'm going to go back. I'm going to do edit, stroke. Let's try 12 pixels. This is pretty good. So this is commonly the type of thing you're going to see in a published paper in terms of black and white DNA gel type data, something like this. Now, of course, there are infinite sorts of types of customizations that you can do surrounding the data. So a very, very common thing is people will mark off where the lanes are. So let me show you. If you wanted to do this, what you might do is you might construct a highlighted box, and ideally your box, you want it to be the same size as that edit stroke, about 12 pixels. And then you fill that in. This is on a new layer. And then now you can actually mark off the lanes. See what I'm doing here? And if I wanted to, if I was really doing this really cautiously and consistent, what I would do is I would actually just make one of these lane markers, and then I would copy paste one to align them with the lanes. And the reason I would do that is because it would be exactly the same length. But this is sort of, I guess, a quick way to do it. Perhaps it's pointless to demonstrate the most inefficient way. Okay, so now we have a separate layer which marks where our lanes are. Now you can see here exactly what I was talking about. This lane is a different length marking than this lane. So this is actually like not the best way to do this. Again, the best way to do this is take one, edit, copy, edit, paste, and then you can just make one and position copies of that one where it goes, and then they would all be the same, if that makes sense. That's one way to do it. But again, I'm just demonstrating this quickly. Now, another thing that some people might do is they might actually crop out the ladder, and you can crop out the ladder if beforehand you mark where these ladder positions are. So sometimes people will make a layer. Oh, let me bring it on top. And mark off where each of these positions are. You can do this edit, copy, and now paste. And then I can pull this marker to each of these ladder positions. And if you have trouble getting it perfect, you can just zoom in. And I would mark off where each of these are. Okay, and then once you had marked off where each of these are, it would be okay for you to crop out the ladder because you've marked that information. So you'll also see that very, very commonly. And then you'll also see people label the lanes. So just quickly, the font you always want to use when labeling figures for papers is an Arial font. And the reason is because Arial is generally accepted as a font by almost all journals. So it just makes it easy on yourself if you always use Arial font. And I'm not going to do anything complicated here. Let's just do 1, 2, 3, for example. You would label these. And I would spend time positioning them perfectly. You know what I mean? It might be, one way to do this might be to correctly put them on a spacing, or you might put your 2 as a separate element to arrange it perfectly where it goes, etc. You might put 4 as a separate element to arrange it perfectly. This is lane 4, where it goes, etc., etc., etc. Okay. Now, one thing I would tell you, since we're here, since we're doing this, one thing I would tell you is the size of your font should be the same size as the sort of line elements you're adding. So the stroke was 12 pixels around this image. Your font, you should try to get it to match that. Okay, so 72 bold. Let's try that. This is going to look much better. Something like this is going to look much better in the end. Because it's much more close to this. So this goes to that critique, which again applies here, where the original newbie mistake is you're importing elements from different universes. Text is often an infringement of that rule. Importing text here is sort of like a strange element that doesn't visually make sense. But if you can make the text look like it fits within the image, it's generally going to look much, much, much better. So I try to always have my font be a consistent font, Arial, and I try to make it such that the pixel thickness of those elements actually matches the pixel thickness of my stroke around the gel data. Now, nothing that I've done here has influenced any of the data inside of the box. All that I've done is actually make the data clearer and easier to interpret, and it just looks better. It's more pleasing on the eye. So this is usually the classical treatment that people will do with DNA gels.